Hello people, welcome back once again to another video of Scientific Monday. Today we have a breakthrough in the genetic world. The source of this is EurekAlert.org and Salk.edu. The links will be down below as usual. So what's so new about this technique? They actually made blind animals, blind mice, able to see again. This is a real breakthrough because the original, more traditional idea of gene editing doesn't allow for stuff like this, because the cells that are in your eyes, when they die off, they get replaced by a new one, but in the meantime they don't divide anymore. So that's the same for the brain and the heart area and your eye. So they found a way to still insert a piece of DNA while the cell is not dividing. So let's go to the article, shall we? So the Salk Institute researchers discovered the holy grail of gene editing, the ability to insert DNA in at a target location in a cell that does not divide. So that's actually the major of adult tissue and organs. Best examples for this are your heart, eyes and brain, like I said before. So this technique allowed them to restore the visual responses of blind rodents, mice to be exact. But so this research will open up the possibility for new research and new experiments where we can make blind people see again, or make deaf people hear again if the problem is at a location that we can, if the problem, so to say, is at a location that we can target it with the technique. So the team is very excited by the technology they discovered, because it's something that could not be done before, as said by June Carlos Espiritua Belmont, a professor at Salk's Gene Expression Laboratory and senior author of the paper that has been published at November 16, 2016 in Nature. For the first time, we can enter into cells that do not divide and modify the DNA at will. The possible applications of this discovery are vast. Certainly, they are. So they talk about the new technique that they call NHIEG. I'm bad at English letters, at least saying them in English, that sucks. So that stands for non-homologous end joining. Damn, that's complicated for me. Not used to this. So it basically repairs DNA breaks by rejoining the original strands of DNA at the point where they broke. Once paired, in the pairing process, they are able to insert their own DNA that they want to have in there, like the DNA that was corrupted and caused the mice to go blind. They invited the gene that they needed to see into those cells. The DNA repaired and the mice can see again. So it's a revolutionary for genome editing in living adult organisms, says Karichiro Suzuki, a senior researcher associate in the Espiritua Belmont lab and one of the paper's lead authors. No one has done this before them. Just to make it clear, this totally new, unexpected. They improved the vision of blind rats quickly this way, and they see capabilities and possibilities to also do this at humans. So I'm going to skip this bit. So we now have a technology that allows us to modify the DNA of non-dividing cells to fix broken genes in the brain, heart and liver, says Espiritua Belmonte. It allows us for the first time to be able to dream of curing diseases that we couldn't before, which is exciting. It certainly is. And the limiting of this application is only limited by what we are allowed to do by government bodies, so to say, because you all have a certain problem with ethics and gene editing, but things like this are amazing. This is where the future actually is, and this is what we can do on a daily basis if we can work this out further. Now on the Salk page there is basically the same information, a photo of the team added to it, a photo of the research, as the description down below says. The cell nuclei, the core of the cell is blue, and the edited genome, that's the green fluorescent part. You can see it's pretty good. Also a clip where the team themselves explain a bit about the new technique, how it works, and the thoughts about it. So go watch it if you want to. Link will be also down below. This is a very short, very quick Scientific Monday, the way I prefer to keep them, not too much in depth, so you can do your own read up if you want to do, and I encourage you to do that because you learn so much more from reading it yourself than just listening. So that's it for this Scientific Monday, guys, already. Thank you for watching. Subscribe down below, leave a comment, like or dislike this video, whatever, and see you guys next time again. And once again, thank you for watching.